Hello, everyone. We are talking about the cell this week, so we're getting started. I'm going to open up my slide. Well, I'll actually leave them like this. I hopefully let me get a bit bigger here. There we go. All right. So I've already given you the slides. They are in Canvas. So this week we're talking about this cell, and obviously we're not talking about every single cell in the human body because a lot of them are very different. They act different. They do different things. So as we get into other um, systems in the body, we'll talk about those cells individually if they're different. So here's our objectives. I won't read through all those. You can read those yourself. So we're going to start with the functions of the cell. So the cells have 8 functions. Now, they don't all have the same functions. Some of them are a little different, but they all have the same, or I shouldn't say they all have the same. They should have one or more of these 8 functions. So the first one is growth. So if you have tissue growth, that's cells dividing, expanding, those kind of things. Reproduction, so that would be the cells that are your gametes, so that you can reproduce another human being. Response to stimuli. So many of the cells respond to any kind of particular stimuli. Metabolism, which we'll go over in just a minute. We'll go over that more even later. Homeostasis, so it keeps your body in check. Make sure that everything is working properly. Movement, protein synthesis. This is a huge thing the cells do, and we're going to talk about how important proteins are to the human body. And transport of substances across the cell membrane. So this will keep your electrolytes, your water balance, all of those things in a nice homeostatic environment. So let's just start with the cell structures. So you don't need to know every cell structure here. Some of them have different functions, I would say, that don't really pertain to what we need to know. So we're just going to kind of skim over these. The one thing that I don't talk about too much in this lecture, but I will bring up, is you see these secretory vesicles. So as things are caught inside of the cell, and the phagocytes will actually envelop around those abnormal or what should I say, foreign bodies that are within the cell, and then they go to the cell wall, and you have the secretory vesicle where they're expelled out of the cell. So let's start with the big guy, the nucleus or the nucleolus. Now, nucleolus is inside of the nucleus. It is not present in every cell. For example, in mature red blood cells, there's no nucleus. Now, when red blood cells are first made, they're called nucleated red blood cells, or NRBCs. And that means that they're immature, that they haven't lost that nucleus yet. So they're not functioning red blood cells until the nucleus is gone. So they look very similar to a white blood cell when they're nucleated. So if you look at a complete blood count or a CBC, you might get a number, and then maybe later that'll correct it, and they'll say, maybe I'll say like 34,000 white blood cells. And you're like, oh, wow, that's really high. And then it'll come back and it'll say 12,000 red or white blood cells, all of these nucleated red cells. So they counted them as white blood cells, but they're not white blood cells. They're red blood cells with a nucleus. So you might see that. That is a sign that the red blood cells are getting used up and the bone marrow is making more. So you have a lot of nucleated reds. On a normal CBC, you might see a couple, maybe, but not very many. The nucleus is the largest organelle, typically, It is, oh, I just told you about the correction on the CBC. So nucleolus is located within the nucleus, and this is where usually your RNA, your DNA are located. Not always so. Sometimes it's just in the nucleus. And then we're going to talk about the mitochondria here. So the mitochondria, we always call is kind of the powerhouse. This is my little fun mitochondria down here. It's the powerhouse of the cell. So it is making energy. So we call it cellular respiration. And why we call it respiration is that it takes oxygen to make energy and it releases carbon dioxide. So respiration is the exchange of oxygen and carbon dioxide. And energy, it produces energy. And we're going to talk about that in a bit later in the lecture. Disorders of the mitochondria cause muscle weakness, loss of muscle strength, nervous system dysfunction, heart block, seizures, and why? Well, obviously, if they're not functioning correctly, that means that you don't have the energy. And they're not going to work properly because it doesn't have the energy it needs to work properly. I had attached a video in the module on mitochondrial disease. It's a relatively newer pathway we look at disease. So if you have somebody with chronic fatigue, it's just not getting better, and you've tested for everything else you might think of mitochondrial disease. It is not the first disease that you think of when someone comes in with fatigue. I mean, there's so many other things that are more common than mitochondrial disease. But I thought the video was very interesting, kind of tells you a little bit about the mitochondria and the history of mitochondria. They think is that it's a separate organism that worked synergistically with our body, and that our body just absorbed the mitochondria as part of our body. Mitochondrial DNA is different than your other DNA. So that's always interesting. Mitochondrial DNA, you can actually follow like a pathway in your family. So a lot of times they'll use that to maybe look at previous ancestors. And I'm talking like way back ancestors. So mitochondria, very, very important. The Golgi complex is what it does is it refines and directs transport of synthesized proteins. So why are proteins so important in our bodies? So you say, well, I need protein to build muscle. You need protein to build almost anything in your body. It's required for enzymes, hormones. I mean, other parts of the cells are made with protein. Protein is so important to the human body. And that is what DNA makes is proteins. So we know how the amino acids attach to the DNA. So if your protein is not being absorbed properly, made properly, these can all cause diseases. So it's very important to our bodies with the proteins. So the Golgi complex, it refines and directs the transport of when we make proteins. So there are some structural changes to the Golgi complex that cause different types of diseases. And this means that you're not making the protein right, or it's not getting to where it needs to go. So structural changes are associated with neurodegenerative diseases, several of them. ALS is one of them, Alzheimer's, Parkinson's, and Huntington's. Those all have to do with the Golgi complex and not working properly. We'll get into those diseases a little bit more when we talk about the nervous system. So the rough endoplasmic reticulum, this synthesizes and transports protein and lipids components of the cell organelles. And it's sensing cellular stress. So when there's cellular stress, it causes more of these proteins to be made. So we talked a little bit about why proteins are so important, but they're important too for transport and communication of within the cell so that the body knows what it needs and it can produce what it needs. Ribosomes are what are sitting on the rough endoplasmic reticulum. That's why we call it the rough endoplasmic reticulum. And if you look here, a ribosome makes the RNA protein complexes. So it makes proteins. So your DNA will separate and you will get RNA. And we have messenger RNA here, which is saying, I need this particular protein made for my body. So it basically DNA unzips and you have the messenger RNA here, and the ribosome is facilitating this binding of amino acids that are coming here, or it's releasing amino acids so that whatever you need is put on that zipper of the messenger RNA to make the protein that your body needs. So this is peptide synthesis or protein making. Disorders of the ribosomes can cause cellular hypoproliferation, which means the cells or that protein's not being made. And that can cause different disorders like anemia. You're not making the hemoglobin, that protein hemoglobin for the red blood cells. Or you have bone marrow failure or even cancer. So those cells are making the wrong protein and causing different kinds of disorders. The smooth endoplasmic reticulum this is where we synthesize steroid hormones or we remove toxic substances. So they don't have the ribosomes like the rough endoplasmic reticulum. The lysosome, it destroys defunct organelles or intracellular digestive system. So any little pieces of something in there is going to be picked up by the liposome and then it is removed from the cell. Liposomes or lysosomes have a newer outlook. The lysosome wasn't getting its due, but now it gets its due. It actually is tied to quite a few different disorders. So first of all, what happens as a person's lysosome age, and how does this affect their overall health? So the lysosome will increase in size and number, but they don't do as much as they used to. So things kind of accumulate in the lysosome. So proteins are degraded and unable to clear that defunct organelle. So you got some junk sitting in the cells. So the lysosome should have been removing that, but it's not really removing it, and the cells are not working properly. Disorder of lysosomes, and these are called lysosomal storage diseases, can cause a buildup of toxic substances and can actually cause cellular death, hence death of the person if it gets really, really bad. I gave a couple of videos on Niemann-Pick disease and Tay-Sachs disease. So these two diseases, you'll kind of look at the videos, but both of them are lysosomal storage diseases. They are neurodegenerative, which means it is a relatively slow, horrible process before death, that losing some of the function of the neurons. So what holds all the cells together? So that's the cellular membrane. So all the organelles are inside of the cellular membrane. This is not the best picture, but I like this picture because it kind of shows you some of the things that sit in the cell membrane. So you do have these glycoproteins and they have carbohydrates attached. And the carbohydrates make it kind of a slick surface so that cells, when they're sitting next to each other, move smoothly, that they don't get stuck, and then the tissue doesn't move like it should. So those glycoproteins help with that, and glycolipids as well. They will have these carbohydrates stuck here. So the carbohydrates is making it a slick surface. You also see some channels here. So these channel proteins, and that allows for the exchange of different ions or fluid. You also see some of these peripheral membrane proteins and integral membrane proteins, and this allows for things to attach to the cellular membrane to either deposit things within the cell or move things out of the cell. So what we know about the cell membrane is composed of that lipid bilayer. So it isolates the microdomains, which differ in protein and lipid composition. It has membrane-associated proteins, and it has a transmember protein, which goes across both bilayers. These layers require lipids. So, when people say, I'm gonna never eat fats, well, that's near impossible because fats are in a lot of different foods. But you need the lipids to create different things for the cells. So it's not a good idea to get rid of all your fats. So lipids, you have phospholipids, and they repair the membrane. Cholesterol, we already saw, is making the cells glide better or move across. Glycolipids are the same. And these arrange in a hydrophobic, hydrophilic membrane. So if you go back here, you'll see the two layers here. There's a split down the middle. So for that, we have hydrophobic on the inside, and hydrophobic means it doesn't do well with water. So it's on the inside, keeping that membrane together. Or the hydrophilic that does like water is on the outside, so water can be there, but it doesn't cross the membrane very easily. Proteins, there are 20 different types of amino acids that are needed, and we need these for cellular transport. So if the cell is moving, we need these types of proteins. Oops, sorry. And carbohydrates, these break down into sugars, but they will give a carbohydrate train, and they're located on the outside of the cell membrane, and they protect the cell membrane and cause lubrication so it can move very easily. So what holds all the cells together in tissues? How come they're not just floating around? Well, we don't want that in some places, right? So that is the extracellular matrix, or ECM, and it functions as a mechanical support to the tissues, but it does keep them in check. So you don't have cell proliferation out of check where you have tissue growing, and nothing stopping it. It does form the scaffold for tissue regeneration. So if you injure your skin and there's a break, then you can, you get that scab and everything. That's your extracellular matrix is being placed so that cell regeneration can take place there. It also has tissue microenvironments. So all our tissues don't all blend into one big mass. We have different tissues sitting next to each other, and the extracellular matrix keeps them from blending together. And what is it made of? I have Jello down here, but it's not made of Jello. It's made of collagen. So collagen cells bind to the desired shape of the tissue. So if you have something wrong with collagen, it affects your tissues. It has elastin, which gives tissues elasticity and flexibility. If we didn't have elastin, then the tissue would rupture or become damaged very easily. Fibronectin, you might see this. This is a breakdown of certain tissues, give off fibronectin. Sometimes you'll order fibronectin levels, and that means there's some kind of breakdown of this structure. I work in the newer intensive care. I know that OB, they order a lot of fetal fibronectin, and that's looking to see if you're going to deliver soon. So it's it's the lab that is checking to see if that fibronectin from the fetus is there, then the mother's going to deliver. It kind of helps with whether we determine if they're in preterm labor or not. So all cells have to work together. And so how do they talk to each other? How do they communicate? They do this by signal transduction, not a cell phone, but signal transduction. So there's 6 different ways of signal transduction for cells. They can be contact dependent. So this requires their membrane to have a signal molecule. And if something attaches to that signal molecule, it sets off a flow of ions. So an example would be gap junctions in the cardiac muscle. This controls the flow of ions in a rhythmic pattern. So these are charged particles. And in the heart, there's electrical activity, and it will send a electrical charge through the heart muscle. And that is done by contact dependent. So that's where you have these signal molecules in the membrane. Paracrine is where the cell secretes to a target cell. So this is an example would be tissue repair. If we have injury to a tissue, it will release certain chemicals, and that will send these other cells to come to the site. So like the white blood cells, and then all of your fibrinogen and all of those will come to start to create a scab or scar tissue, basically. Autocrine, this secretes to itself. So T cells are used in immunization. They are activated in this way once they kind of trigger themselves to start to go to the site of inflammation. Hormonal, this secretes in the bloodstream to a target cell. So one of the ones that we all know is thyrotropin, and that stimulates release of thyroid stimulating hormone. You might have neurohormonal, and this secretes from the neuron to the target cells through the blood system. So we think of oxytocin, and this, causes the uterus to start to contract. And then we have neurotransmitters. They secrete from a neuron to a receptor cell. Dopamine is one of those. Any of those neurotransmitters that occur in the brain, for dopamine, for epinephrine, for norepinephrine, all of those are an example of those types of communications. Cellular adaptation. So our cells will adapt to different stimuli, and there's several different types. So the first one is atrophy. This is the cell decreasing in size. Usually it's loss of function. So we think about this in maybe somebody who's had a spinal cord injury, and they're no longer able to use their legs. So the muscles are no longer moving. And so they atrophy. They start to get smaller because they're not functioning. It could be caused by starvation also, where it's not receiving the energy it needs, or ischemia, lack of oxygen to the site, or pressure or forces as well can cause atrophy. If you look at a brain for a person with Alzheimer's, you'll see the brain atrophying. It gets smaller, and so you see these wider gaps between the sulci. Hypertrophy versus hyperplasia, they are not the same thing. So hypertrophy means that they're getting big. So I like this picture, and that these are the cells. Hypertrophy means that the cell is now bigger. Hyperplasia means you have more cells. So hyperplasia means instead of just these three, now you have these 5 cells. So they're not interchangeable. Hypertrophy means that they're enlarged. Hyperplasia means that there's more of them, that it's growing in size based on more cells present. Just gonna move my slides a little here. Metaplasia versus dysplasia. Here's another good There's another good picture for that. So for metaplasia, the cells are replaced with different types of cells. So this can be in a healing process. So if you have an injury to your skin, the cells will come back and they'll replace it, but they're not the same kind of skin cells as maybe that were there before. So they can't grow hair. They may not have sweat glands, but it looks like skin, but it's not the same as the previous skin. It acts a little differently. So if you look down here, metaplasia is conversion of one cell type to another. Dysplasia means it's abnormal growth, size, shape, or organization. It is not normal growth. So you see here, it's very disorganized. So that's the difference. This is replaced with one type of cell. This could be all different kinds of cells. Necrosis versus apoptosis. So necrosis is cellular death due to swelling, and it's usually due to exposure to some substance. So if you look here for necrosis on this side, on the right side, you see cellular swelling. And this could be due to the cell taking in more water or taking in more electrolytes that pulls in more water, something like that. And then the cell membrane ruptures because it only holds so much. And because of this rupture, it releases the intracellular contents into the extracellular space. Apoptosis is a loss of cells due to shrinkage, and it's regulated by genes. So here the membrane is starting to bleb, starting to become weaker, and then it ruptures because it's not functioning well. It's kind of, you might think of it as an aging process, possibly. And then it releases these cell fragments out into the between the cells. And then the phagocyte or macrophage or phagocyte will actually pick up all these pieces and then remove it from the tissue. All right, so now we're going to talk a little bit back onto proteins. And so we have proteostasis, and this is the regulation of proteins in the cell. So we need proteins to be made. It's very, very important, but we don't want them getting out of control. So this helps the person adapt to the environment and prevent disease. Understanding proteostasis helps with understanding the treatment for some diseases. So there are 4 major proteolytic cascades, and we're going to go through them. Proteases are enzymes that break down proteins into smaller pieces. So we call that proteolytic cleavage. So the first one we're going to look at is the caspase-mediated apoptosis. So we already know what happens with apoptosis, right? So these capsases dismantle cells for tissue homeostasis. So maybe the cell isn't functioning well, it's old, it needs to go, it needs to make room for new cells. So in this, it receives a stimuli. So here you see the apoptotic stimuli, and that goes into the cell. And then it breaks down, like it says. And then this capsases will break it down even farther. And so we have true apoptosis. This allows for cell growth without hyperplasia. So we're getting rid of old cells, we're putting in new cells, but we're going to have pretty much similar number of cells. Now, you're probably asking, do I need to know every step of this? No, you don't need to know every step of this. What you need to know is apoptosis, what it is, And capase helps with that in healthy tissue. So this is not pathologic. This is normal tissue regulation or homeostasis. We're getting rid of tissue that is not working or having issues, old tissue, and replacing it with new cell growth. So that is normal. Blood coagulation, it's where we stopping bleeding, right? So it's changing blood from a liquid form to a gel form or making a clot due to injury. Again, you don't have to memorize all these steps, but you do need to understand there's different phases in the blood coagulation. And we're going to talk about this more later when we talk about hematological dysfunctions, because some of these cascading things that you see here, the different phases, and all these numbers are factors. These factors must be present for you to make a clot. So when we talk about like hemophilia, where one of the factors is missing, it will go through the stage, but it will only get so far. And if let's say factor 8 is missing, then we're not going to have this step. So we're not going to make a clot because we don't have each of these steps that it must go through to change prothrombin into thrombin, and then that changes into fibrinogen, fibrin, and then it makes your clot. So you don't need to memorize these steps, but you do need to understand that, see all these different factors that are involved in this and all the different steps to get to the clot. So if you're missing something or one of these isn't functioning correctly, then you're not going to clot. This is good to know also if we're talking about some medications that prevent you from clotting. Matrix metalloproteinase is, or better known as MMP, are enzymes that break down that extracellular matrix. Remember, that's where all the tissue is built up on there. And this is breaking down that framework. And this does occur in wound healing and tissue remodeling. So if the tissue is injured, it removes all that framework so that tissue can be sloughed away and new tissue can be grown. Now, it can be pathologic. So you could have where the tissue, healthy tissue is removed and not so healthy tissue is replaced. You see this in arthritis. You see this in cancer and cardiovascular disease. What keeps it in check, because we don't want it to keep breaking down the framework, because then, where does it stop? What keeps it in check is the tissue inhibitor of matrix metalloproteinase, so TIMP. So you're like, well, where does this take place? It takes place in a lot of different places. So cardiovascular disease, diabetes, renal pathologies, gynecological and obstetrics diseases, cancer, inflammatory, and neurodegenerative diseases. Now, you don't need to remember all of these different types of MMPs or the TIMP that stops it. What you just need to know is that all of these diseases are caused by this process with the MMP. So there's something out of the homeostasis isn't there. Something is causing tissue damage. And then the last one is the complement cascade. This is a cascade of enzymes crucial for the immune system. This is an amplification of the immune system. It destroys pathogens and clears away cellular debris. So again, here you'll see this pathway. It's got a lot of different things going on here. You don't have to memorize all of these, but you do know that this kind of cascade takes place in cell lysis, so killing of pathogens. So it lyses bacterial pathogens, those kind of things. Opsonization or phagocytosis, this clears away that debris. And regulations inhibits of the complement cascade, so it stops all of that breaking down. Other functions, promotion of cell differentiation multiplication and recruitment, modulation of immune cell migration, regulation of the beta cell functions, and leukocyte adhesions. So how do we get something across the cell membrane? So movement across the cell membrane depends on what it is as to how easy it is to get across or how do we stop it. So we have a gated transportation recognition and binding receptors. So as whatever substance is coming, if it binds to the receptors and the cell needs it, it will pull it in. If it doesn't need it, it won't pull it in. Vesicular transport. So a vesicle forms inside of the cell. It goes to the cell membrane. It will part the cell membrane. And then if anything needs to come in, it will fuse around that and pull it in. Or it'll go the opposite way. So if you have something in the cell you need to get rid of, it will engulf around that, go to the cell membrane, open the cell membrane, and out it goes. Transport channels, you saw those on that slide with the cell membrane. So there are some things that freely cross the cell membrane depending on what the cell membrane is, that particular cell is for. So it just depends. Some things cross against some cells, but not all cells. Active pump, you'll see this especially with electron or electrolyte transfer. So remember, electrolytes are charged. So you're moving ions or different electrical states of these elements across the cell membrane. ATPase is required for sodium-potassium transport over cell membrane. Remember, potassium sits on the inside of the cell, sodium sits on the outside, but there is a balance. Sometimes you need sodium into the cell or potassium out of the cell for that balance. But remember, potassium sits inside a cell and sodium's outside the cell. Calcium is also moved by an ATP-dependent method. Which muscle do we worry the most about with alternation of calcium? And this is the muscle cells, or especially the heart muscle cells. So we always worry about our calcium levels when we have heart problems. Another way to move things is passive transport or diffusion. This is moving the particles. So the rate depends on the charge of the particles. So if you are at a difference of charges, then it's easier to move. But once the charges equal out, it's very difficult to move a charged particle. But diffusion is the movement of particles. And diffusion means that it goes from the high concentration of particles to the low to equal out the number. Another passive transport is osmosis, and this is the movement of water, not particles, but water. So water is moving, and it goes from a low concentration to a high concentration. So you kind of have particles moving the opposite direction water is till it's at a homeostasis. Now you do have filtration. This is pushed through by a force. You think of this with your blood pressure, that force coming from the heart, pushing blood cells to where they need to go. Also, and it also pushes substances into the tissues. Endocytosis and exocytosis, this is where endocytosis, you're engulfing a substance within the cell and then moving it outside. Exocytosis is moving from outside inside. Cellular metabolism. So I'm not going to spend a ton of time on this because we're going to spend even more time on this when we go over digestion and metabolism of different foods. So first of all, we have to have digestion. So this is where we're eating foods in our mouth, our stomach, the intestine breaks them down into subunits. So that's what digestion is, breaking it down into subunits. Carbohydrates changes to glucose. Proteins change to amino acids. Fats change to fatty acids. But like I said, more to come on that. The breakdown of glucose to energy, though, is when, and this is just one of those pathways. Remember, we said carbohydrates goes to glucose. Glucose then goes through glycolysis, and it breaks down glucose to pyruvate, and pyruvate then releases ATP. So when that pyruvate comes into this cell, it goes through some stages, and one of those is the Krebs cycle. I don't know if you remember that back when you took physiology, or even in high school, maybe you took some science courses and remember the Krebs cycle. No, I'm not going to make you memorize the Krebs cycle. It's got several stages to it. But the thing to remember about the Krebs cycle is it's aerobic, which means it needs air. You need oxygen. So one molecule of glucose going through the Krebs cycle, which is aerobic, gives you 38 ATP, and that is your ATP that you need for energy. Anaerobic metabolism does not use oxygen. You think, well, that's great. So if my body's still going to work if I don't have oxygen, well, it's not going to work very well, because that glucose going through the anaerobic metabolism will only give off 2 ATP. So not very much energy at all. But it also gives off lactic acid, which leads to metabolic acidosis. So that's a huge issue. Again, we're going to talk about this more later, but you do need to know the first steps of this for this first lecture. Cellular destruction and injury. So what happens when our cells are injured or they're destroyed? So cellular destruction can be caused by many things. First of all, bacteria. It can destroy the membrane and hence the cell breaks apart, or it affects the cellular division. Viruses are not, there's always a debate on whether viruses are a living thing or not. The thing with viruses is that it attaches to the cell membrane, and it injects the genetic material inside the virus into the cell. So your cell will replicate the DNA of the virus. So it's basically creating more virus. And it's using up all the energy the cell needs to make all these little pieces of DNA or all these extra viruses. Then the cell ruptures and it releases more viruses out into the other tissue. So a virus cannot live outside of a living being. It can, what should I say? It'll be present, but it's not living until it attaches itself to a cell. And then that's how it replicates itself. Chemicals can break down the cell membrane or destroy the organelles. Physical destruction, injury to tissues will cause cellular destruction. And then underlying medical conditions can affect the cells as well. So if we think of sickle cell, which we'll talk about sickle cell a little bit later, but sickle cell is affecting what the shape is of the cell, the red blood cell. And because it's misshapen, it doesn't bind or doesn't have a good affinity for oxygen. So the tissue becomes hypoxic because there's no oxygen being delivered because of these red sickle celled RBCs. Cellular injury can be caused by ischemia or hypoxia. So decreases oxygen to the cells. It goes into anaerobic respiration, causes acidosis, which causes tissue destruction. But sometimes if we have ischemia, which is decreased oxygenation to the tissue, and then suddenly we give it a bunch of oxygen, we can cause a reperfusion injury. So oxygen in itself is a free radical, and it can change shapes of cells. It can change how the cell functions. So it's not, gosh, back in, I don't know if it was 80s or 90s, they had those oxygen bars where people were just breathing 100% oxygen. They were like, oh, I feel so good. And it's like, why? Because what you're doing is causing more tissue injury. Sometimes hyperoxygenation can be just as destructive as low oxygen levels in the cell. So it's not something we strive for. We don't like to provide too much oxygen because it will cause damage to tissues. One of those tissue death from too much oxygen we see in our babies. So if we give too much oxygen to the term babies, it can affect their bone marrow, and later on they might develop leukemia. So it's not something we should just be freely giving to anyone. It's just something that we need to keep in control so that we are making sure we don't have hypoxia. So we'll make sure we're getting oxygen to the tissue. But remember that reperfusion injury, if we give too much oxygen, it can also cause damage. Chemicals such as toxins, these can cause changes to the DNA. And this is called epigenetics. And so we're going to talk about that next week, about genetics and how it can affect all these chemicals that can affect our DNA. So we'll actually change the structure of your DNA. And some of those are air pollution, exposure to lead, mercury, cadmium, arsenic, or even drug use or ethanol. And then, of course, injury can cause injury to the tissue. So we have infections, accidents, physical agents, such as climate changes, radiation, or mechanical stress. All right, so that's the end of this lecture. So I will be posting this. Again, I kind of went over what you should and shouldn't study for the quiz. As far as those pathways, you don't need to know what step, which one's which step, but you do need to know what's happening in the pathway. All right, if you have any questions, you can put them on the discussion board, that question part when you first open Canvas, and I will try to answer them, or you can save them for a review session. That is fine. All right, you have a good day. Bye.